Thank you. And I appreciate the opportunity to be here and tell the story on behalf of all our employees, our stakeholders, and of course, patients around the world. I didn't come alone. I have our CFO with us today, Jean-Pierre. If you want to get a different perspective besides mine, feel free to talk to him. This is my disclaimer. We were bought by Cordis, so these things that I'm saying don't necessarily represent the views of Cordis. They're my own. So I've done 15 startups. 3 were IPOs, one on NASDAQ, one on New York Stock Exchange, and one on the XGX in Singapore. And I've had 10 trade sales to 7 different companies. Boston bought 3 of them. So, and you might recognize some of those names, but today I'm here to talk about the last one, MedAlliance. So this is the problem we're trying to solve. Arteriosclerosis, and this is a disease that starts very young. And we learned this from Vietnam soldiers who we operated on at 18, 19 years old, and we saw the beginning of arteriosclerosis. So this is a lifelong disease. It certainly affects the heart, but it affects every vessel. The only reason it affects the heart the most is because they're small vessels. And everywhere there's small vessels, including in below the knee, this is where you first get problems. So we're based in Nyon, Switzerland, which is between Lausanne and Geneva. We have R&D in Irvine, California. We have a regulatory group that's in Scotland, and we have some advanced R&D in Singapore, and that's because my CTO lived in Singapore. We're privately owned. We were born global, virtual to begin with, and almost the entire team were people that I'd worked with before. And many of them not only in biosensors. John Schultz was a CTO. I was a CEO for 13 years of biosensors. But we had people that I've worked with in 3, 4, in one case, 5 different companies. So here was our vision. I was CEO, as I just mentioned, of biosensors for 13 years, and we made drug-eluting stents. And because you're collecting data all the time in drug-eluting stents, we saw where they didn't work so well. They didn't work well in long lesions. They didn't work well in small diameter lesions. They didn't work well when you had a bifurcation. They didn't work well in patients that had high bleeding risk. And we know that with permanent metal implants, there's a 2 to 3 complication rate, 2 to 3% complication rate per year. So that's 10 to 15% at 5 years. And we also started noticing that other DES manufacturers started manufacturing stents with thinner and thinner struts, which meant they didn't really have much radial strength. And we thought, well, if you don't really need radial strength, then why have a stent at all? So we thought you could put drug on a balloon and deliver it without leaving some metal behind. And when we started, all the balloons that were on the market were using a drug called paclitaxel, which is a cancer drug. And we at Biosensors had a paclitaxel drug-eluting stent, and we had a limus drug-eluting stent. We eventually took the paclitaxel one off the market because the performance of the limus DS was so much better. Late loss was always about half of what it was with paclitaxel. So we thought we had an opportunity where we can improve patient outcomes. And then we saw that DCB at that time was pretty much only used in peripheral artery disease in the leg, in SFA. And we thought that there were many other applications, primarily coronary. And at that time, nobody was really looking at coronary. But it's basically, as I said, this disease is systemic, so it's everywhere. So it's for ED, it's for AV fistula, it's for neural, it's for renal. We saw lots of applications for this platform. So why is a drug-coated balloon better than a drug-eluting stent? Well, the first reason is it reserves physiological function. And what does that mean? When the heart beats, the vessel expands and contracts with every heartbeat. If you have a cage there for a stent, it can't do that. It also allowed for positive remodeling, which meant over time, the vessel could get bigger, whereas with a stent, it only gets smaller. And then we avoid these complications I mentioned with metallic scaffold, which is an inflammatory response, but also sometimes they fracture. And then the overall thrombosis and other complications that you get, as I mentioned, was between 2 and 3%. The other advantage is if somebody has a drug-eluting stent, they have to take 2 pills every day. So they have a dual antiplatelet therapy. So every day for 12 months, these people have to take these antiplatelets, which also makes you bruise more easily. I mean, there are a lot of disadvantages to DAPT. But with our drug-eluting balloon, you only need 1 month, not 12 months of DAPT. Plus, you did all this and you could still put in a stent later or have surgery. So it didn't prevent you it kept those options open later if you decided you wanted to do that. These are the studies that showed that drug-eluting stents have a complication rate of between 2 and 3% per year, which is 10 to 15% in 5 years. So why sirolimus over paclitaxel? Well, sirolimus is cytostatic, where paclitaxel is cytotoxic. What does that mean? Paclitaxel is a cancer drug, and it kills all cells indiscriminately. Sirolimus, on the other hand, just prevents them from reproducing. And the other big advantage of sirolimus is it's an anti-inflammatory, where paclitaxel definitely is not. The margin of safety and the therapeutic range of sirolimus is well published. It's many times greater than paclitaxel. They both work as anti-restenotic drugs, but as I mentioned before, the weight loss was always less with sirolimus-based drugs, and it had this big advantage of being an anti-inflammatory. So these were the studies in drug-eluting stents that basically killed paclitaxel. You had half the in-stent restenosis rate, you had half the weight loss, you had half the TLR. TLR is the amount of patients coming back for another intervention. So half as many patients had to come back for another intervention. These were all the applications that I mentioned earlier. But based on this, we did our first round, and since we were fortunate enough to do an IPO with biosensors, we basically were able to self-finance the first round. And we used this to do the preclinical animal test. Now, if Limus sounds so much better than paclitaxel, why are there 30 balloons with paclitaxel and there were no balloons with sirolimus? It's a really good reason. It's hard to work with sirolimus. It takes a long time for it to get into the tissue, and it has a short half-life, 82 hours. So if you don't have something to sustain it, it'll be gone way too quickly. So here's our design goals. We wanted to make something that would stick to the balloon and very quickly transfer to the vessel wall, because we wanted to go in coronaries, which means you can't take 2 minutes to transfer the drug. You got to do it in 30 seconds. Most importantly, we needed something that would elute sirolimus over time until you could get healing in the vessel. So this couldn't be 82 hours or 14 days. This had to be 60 days or more. And then we also wanted a coating that would protect so you wouldn't lose it before you got to the lesion. So the idea we came up with was taking a polymer and a drug, like we did with the drug-eluting stent, and also Biotronics did with the drug-eluting stent, and Boston Scientific did with the drug-eluting stent. You make a mixture of 50% drug and 50% polymer, and we put it in a microsphere, a microreservoir, the same size as a red blood cell, 4 microns, so it could pass through the capillaries. And then we had to solve the problem how to get it on the balloon, have it stay there, and have it transfer. So we created this cat membrane technology, which actually took us about 4 years to develop. The microspheres we did in a year, but this cat membrane took a while to develop. And why is it special? It works in 3 ways. First, mechanically, as you press the balloon against the wall of the vessel, the drug goes in. Second is biological, that it's highly lipophilic. So the coating hates the water in the blood, and it really loves the fat in the tissue. So it's drawn to the fat in the tissue. The third thing we did was we noticed that the vessel wall had a negative electric charge. So we put a positive electric charge in the coating, which nobody had ever done before. And we got electrostatic bonding as well. So those were the 3 ways we could have a fast transfer. In terms of the microspheres, they're really small. And this is 100 times magnification, but this is compared to the 2 FDA-approved products which use crystalline. The problem with crystalline is when the balloon expands, it breaks off like crystals in unpredictable sizes and shapes. And this is why the FDA has to this day not approved a drug-eluting balloon for coronary. And remember, from the very beginning, we wanted to go in coronary and we wanted to go to the US. So how did this work in animals? Pretty well. If we look at 1 hour after implant, we were several times greater than the existing FDA-approved product. And it also lasted longer than any other. Here it's 60 days. We now know with later data it's 90 days. And because it was so effective, we could use a lower dose. This is an example of a rabbit artery that's cut open, so you can see the uniform dispersion of the microreservoirs and the cap membrane inside of the artery. When we tested it, we tested it for how much drug would go into the tissue. This is against the FDA-approved products. The red is what you lose before you ever get to the lesion. So that's washed out before you ever get to the place you want to treat. The yellow is what's left on the balloon when you pull it out. And the green is the reason you use a drug-eluting balloon. That's how much drug is transferred to the tissue. So we had several times more drug transferred to the tissue than the existing products. Based on that, we did our second round of funding. So we had a pre-money value of 20 million. We raised just under 5 million. We got it from a Japanese strategic who wanted us to use their balloon in our drug-eluting balloon. So that's why they gave us the money. And this was enough money to start our first in man. At about this time, 2015, there was the first year drug-eluting balloons were in the US. And guess what? They were really widely adopted. So this market was starting to explode, and you see Lutonix, which was BD Bard, Becton Dickinson, Impact is Medtronic, and Boston Scientific. They all jumped into the marketplace. And also in 2015, at a Link meeting in Germany, the most famous pathologist in the world, Rino Vermani, was asked, is paclitaxel, is there any other drugs we're going to use? Because all the drug-eluting balloons were paclitaxel. And she said, tomorrow drugs may change. They may be sirolimus because paclitaxel is toxic. She's the one who just did the animal studies that I showed you, and she just finished them 2 weeks before she gave this talk. So we went into the first in man. We went to Professor Zeller in Germany, who's probably one of the most famous guys in the world in this space, some other German centers, and the results were excellent. We got 0.19 late loss, which is exactly what we got with drug-eluting stents in the coronary. So this was the first time anybody showed you could get the same amount of drug in a peripheral with a balloon than you could with a drug-eluting stent in the coronary. Also, if you remember TLR, I told you before, it's the amount of patients that come back. We were with the lowest amount of patients that come back in any published data to date. We also had low conversion to stents, which was a problem with the early drug-eluting balloons. So based on that data, we did our third round of funding. So we went to 100 million pre. We raised 20 million from a Chinese strategic. Interestingly, this Chinese strategic was the market leader for this dual antiplatelet therapy that I mentioned. And they realized that they're not going to sell 12-month dual antiplatelet therapy as they would with the DS, because with the balloon, you only needed 1 month. So that's why they decided to make the investment. Then we went into where we really wanted to go, which was into the coronary. And the coronary, originally, there was bypass surgery. And then there was this thing called POBEC, balloon angioplasty, which was invented walking distance from here by Andreas Grunzig in Zurich. And the factory was in Bulak, Schneider. The problem with POBA is that occasionally the vessel would collapse. So then the bare metal stent was invented, and that was also first done in Switzerland, in Geneva, by Uli Sigvat. Then the first generation DS came in with a product from Cordis, the company that bought us. They brought in the first DS, the Cypher DS, 20 years ago. Marie-Claude Maurice presented it in Stockholm. Then the second generation DS came, which we were part of at Biosensors, where we went public in Singapore. And then the first generation DEBs came out. This was with paclitaxel. And then fully biodegradable scaffolds, so stents that disappeared over time. That was really important because the physician had to prepare the vessel for that product in the same way that you need to prepare it for a DCB. So that really helped us with acceptance of DCBs. So this was our first coronary trial. It was presented at TCT, which is the coronary meeting in the world in Washington, D.C. And data was presented by Stefan Windecker, who's professor in Bern here in Switzerland. And if you look at the right, that's what the curve looks like before you do an intervention. On the far left is what it is post, immediately after the intervention. And then the follow-up is 9 months later. And what you want is follow-up to look like post, which it does perfectly here. So these were excellent results. Based on that, we went for another round of funding. This time we doubled the pre, 195, and we did a 30 million round, and we did it with another Japanese strategic. And we also received CE mark. So we started building up product for both peripheral and coronaries, and we hired a European sales force with that money. We also went to the US. So this is a time we received 4 breakthrough designations from the FDA. I don't know any company who's ever received 4 breakthroughs, but what this means, and I wish we had a program like this in Europe, which the MDR does not. But with the breakthrough program, you get a fast track to approval if you can show that you improve or extend patients' lives, and you also very importantly, get automatic reimbursement once you reach market. We also received 4 IDE approvals after getting the breakthrough 8 months later. This was key, and it was important because actually these were the first 4 milestones we had to get in the Cordis deal, and we got it the week before we closed the deal. So we already achieved the first 4 milestones. Based on that, we did another $50 million round, and this was to support the US clinical studies. US clinical studies cost about $30 million each, and we were doing 4 of them. So with the breakthrough, we raised another round, this time from bankers in China. Interestingly, we had offers from strategics already at that time, but offers to invest, not to buy. But we didn't want to take a strategic offer because if we took one, it would limit our chance to exit with another strategic. And if they didn't buy us, we would be damaged goods. So even though some of the offers we had for strategics for funding were better, we decided to take a financial offer from CITIC Bank. So then we started these 4 IDEs. I don't know any company that's doing 4 IDEs at the same time. We're in 200 hospitals in the US. These are anywhere from 30 to 80% finished right now. And as I say, these cost $30 million each, so they're not cheap. So we went to Cordis and several other companies, and we asked them for more money. We said we'd like a 300 pre-money. We'd like to raise 35 million so we can do the second and third IDE studies, because we also wanted to go do a study in erectile dysfunction and AV fistula. They thought about it and they said, well, actually, we just like to buy you now. And we said, what number do you have? We said, we kind of have a high number in mind. And so we negotiated, for a bit of time, but eventually we settled with a $1.3 billion with milestones deal. So why did we choose Cordis and not other companies? The reason we chose Cordis was they didn't have a drug-eluting stent to protect. Many of the other companies already had, particularly a coronary drug-eluting stent, and none of them could see that they were going to get cannibalized. And we followed, the old Steve Jobs, if you're going to get cannibalized, do it yourself. So Cordis didn't have a DS, even though they introduced the first DS. We at Biosensors did a study against them and basically showed we were better, and they had to take their product off the market. So they didn't have a DS. They also had a sales force of 900 people. They had access to 10,000 hospitals around the world. They have a whole range of coronary and peripheral products, although none of them were sexy. They had them, and they were selling them. They were selling about $1 billion worth. So this was a complement to that and gave them an innovative product. It also was a source of funding for us. So we manufactured mostly for our clinical studies, but we had to scale up manufacturing. If this was going to be a $3 billion business, we had to scale up manufacturing. They had the expertise and the funding to help us do that. The other thing is, since they just done a buyout and they'd taken it out of Cardinal Health, they didn't have management team and they didn't have a clinical team. So that meant all our clinical team could transfer directly over to Cordis and continue. And that also gave us continuity for our IDZ studies. So that was also an advantage. And as I already mentioned, they didn't have a drug-eluting stent program to protect. So this is just to remind everybody that everybody sees the headline and they think that, we sold $1 billion company and we did it in 2 weeks. We started this in 2008. It took a long time. This is not a quick process. So this is late breaking news. This was presented yesterday in Tel Aviv. We're doing an important study we decided to do, which wasn't for regulatory approval. We did it because we wanted to show a marketing advantage. We're doing a 3,326 patient study, head-to-head against all drug-eluting stents and a drug-eluting balloon. And it's powered for superiority at 5 years based on that 2 to 3% complication rate per year with the stent, and non-inferiority at 1 year. This is from the safety committee, which is headed by Bernie Meyer, who was one of the first guys who invented angioplasty with Grunzik. He's in Bern. And he looked at 2,119 of the first patients at 1 year. And basically he said overall events were rare and they were the same in both groups. So basically this guarantees we're going to hit our primary endpoint. So this was really good news that came out yesterday. So what's next? Well, while we were developing the drug-eluting balloon, we noticed that there was a gap in the market for balloons that prepared for a drug-eluting balloon, because you have to prepare the vessel. The drug-eluting balloon is just used to transfer the drug. It's not used to reshape or get rid of calcium or make the vessel smooth so that it won't restenose. So we created a new company where we're going to focus on making a specialized delivery prep balloon before the DCB. And if anybody's interested in that, we're moving forward. So in conclusion, I'd like to remind everybody that it's not the strongest of us that survive, and it's definitely not the smartest. It's the one that are most adapted to change. Thank you for your time and attention.